SNX-BARs are membrane remodeling proteins that play key roles in human disease. Using our methods, we can determine their precise biophysical properties to facilitate lipid binding and membrane remodeling.
Purifying the SNX-BAR proteins from yeast cells allows the acquisition of native homo and hetero SNX-BAR dimers while minimizing the toxicity and insolubility frequently encountered with non-native expression systems.
Our methods can be used to understand the lipid specificities of all yeast SNX-BARs or to produce recombinant SNX-BAR proteins from any other organism.
A correct extruder assembly is essential to preventing liposome loss during the extrusion process.
For beginning students or those new to the field, visually observing the most critical steps will greatly improve your chances of success.
Begin by inoculating a large swab of yeast cells into a flask at least 4 times the volume of the culture containing 50 milliliters of standard YP medium, supplemented with 2% raffinose and 0.1% glucose as a carbon source, and grow the culture overnight in a 30°C shaker. The next morning, inoculate the 50 milliliter pre-culture in 1 liter of standard YP medium with 2% raffinose and 0.1% glucose in a 2.8 liter volume, baffled Fernbach flask for a 4 to 5 hour culture in the shaking incubator. When the optical density at 600 nanometers reaches 0.2, add galactose to a final concentration of 2% to the cells for an overnight culture in the shaking incubator. The next morning, harvest cells by centrifugation and transfer the yeast pellet into a 50 milliliter conical tube.